So let's conclude now with some practical ideas about how PWMs are used to both characterize TF binding specificity as well as identify actual TF binding sites across the genome.
And so, again, kind of going back to the idea of, well, what can you do with a sequence logo and position weight matrix? So number one, again, you can basically use it to get some sense of the sequence specificity of a transcription factor. And so the sequence specificity of a transcription factor is dictated by how it interacts with both the individual bases that are present in the position weight, or the individual bases that are present at its binding site, as well as the interactions of the TF with the backbone of the DNA as well. And so basically, when you see positions where there's a lot of sequence specificity, that tends to indicate potential, for example, direct interactions between one or more amino acids on the transcription factor to that particular base at that position of the binding site. Whereas sort of less strong preferences for bases could indicate an indirect effect of bases on like the shape or the charge of the backbone, or just might indicate less direct contacts between amino acids and DNA. And so one obvious question is, how good are PWMs for actually finding real binding sites of transcription factors? And so the answer is, it depends on what the context of this binding is. And so if you're just talking about in vitro binding of a transcription factor to, say, naked DNA, the PWM scores actually pretty well correlate with in vitro binding. And so lots of people have done studies trying to measure correlation between the predicted likelihood of binding is via the PWM scores to the actual binding observed in vitro, and found that by and large there's pretty decent correlation between the predicted and actual binding of TFs to DNA. So maybe unsurprisingly in hindsight, it turns out that even though PWMs seem to be able to score in vitro binding sites fairly well for transcription factors, it turns out that they don't really predict in vivo binding sites very well. And so the agreement between like a PWM scan of a genome and, say, a ChIP-seq assay, if you've done in a kind of in a naive way, are pretty uncorrelated. And so it turns out that the in vitro predictions made by PWMs tend to overpredict binding sites of transcription factors, whereas in vivo, most transcription factors don't bind nearly as many sites as that predicted by the PWM. And so this isn't entirely surprising because, as I mentioned earlier, PWM or transcription factors require their binding site to, for example, be accessible. And so obviously in a lot of cellular contexts, a transcription factor binding site might be wrapped up in histones or shut down epigenetically. And so even though there is technically the potential to be bound, the physical accessibility of the genome at that location is just such that the TF won't actually bind there in vivo under many cellular contexts. And so, proponents of PWMs, for example, at first thought, well, I mentioned earlier that PWMs give you a score for determining, essentially what the likelihood of a TF is for binding that particular sequence. And so oftentimes in practice, when you're searching for, real binding sites in a genome sequence, you'd oftentimes set a threshold and say, okay, well, if I see a relative PWM score of at least 90%, then I'll consider that like a real binding site. Otherwise, if it's below 90%, then I won't consider it a real binding site. So some people thought that, well, if you just kind of raised the bar and said, okay, well, I need to see at least 95% or 99% of the maximum score in order to believe that that's going to be a real binding site. It turns out that changing that threshold doesn't really increase your accuracy of predicting in vivo binding by PWM. And so this is in general a really hard problem to address. And so we spent a lot of time during this lecture talking about relative entropy, and its relationship to sequence logos. And so I just want to point out quickly a few key applications where you see a lot of sequence logos and information content and relative entry and so on. Outside of, for example, looking at ChIP-seq data for a particular TF, just to emphasize how prevalent and how important it is to really understand what a sequence logo is. And so, for example, a lot of people look at sequence logos corresponding to alignments of protein-coding genes because they believe that the positions that have a lot of information will tend to point to key residues of a particular protein. So for example, if you have a bunch of homologous copies of a gene aligned and you see positions with high absolute entropy, then that tends to point to really important amino acids that might be involved in, for example, active sites, or might be involved in structural constraints on that particular protein. So for example, it might indicate like a requirement to keep, for example, hydrophobic contacts that are important for the structural integrity of the protein. Sequence logos are also used in terms of RNA sequence analysis, and so we'll talk a little bit about RNA secondary structure, for example, in the transcriptome lecture. But basically, sequence logos are oftentimes used to look at alignments of different RNAs so that you can then estimate, for example, how much entropy or information content there is for a given base in an RNA. And oftentimes, the entropy at a particular position in an RNA can tell you something about the structure of that RNA. And so lots of people have found that, for example, there might be certain motifs of bases near stem loops of RNA that might correspond again to some sort of like functional element or some sort of requirement for maintaining the certain 3D structure of the RNA at that particular position. And finally, sequence logos are oftentimes also used to look across multiple genome alignments to identify, for example, conserved regulatory elements. And therefore, within those conserved regulatory elements, again, they may be used to identify key bases that are bound to by transcription factors. And so here, for example, I'm just showing an alignment of like soybean and rice and and sunflower, for example, where basically sequence alignment and the corresponding sequence logo tells you about the relative conservation of different bases at different positions in the sequence alignment. And again, the general idea when you're looking at multiple genome alignments and using techniques like sequence logos and many other kind of phylogenetic based approaches to look at sequence conservation is that generally speaking, sequences that tend to be conserved across evolution will tend to be functional. That's not always true, but it's a working hypothesis. And so again, sequence logos plays an important role in visualizing where the conserved regions of sequence alignments are.